Okay, I think we can go ahead and start now. Thank you so much for coming. This is about mitochondrial Eve, and I'm Marianne Clark, Max Chatnoir in Second Life, and I teach biology at Texas Wesleyan University. So I think we'll start with what mitochondria are. Is that res for everybody? Cool. Well, mitochondria are the major energy-producing organelle in eukaryotic cells. What they do is convert the energy of high-energy electrons in carbohydrates or fats into the energy-storing molecule ATP, which is the common energy source for most activity in cells, in eukaryotic cells. Eukaryotes are cells that have membranes around their chromosomes. Interestingly, the mitochondrion also has a chromosome. And this is a picture of the mitochondrial genome. It's fairly small. The human mitochondrial genome is very similar to the genomes or the mitochondrial genomes of other vertebrates. There are about 16,500 nucleotides, and the genes are very closely spaced. There's almost no space in between genes. Both of the strands of the genome are represented here, and you can see that most of the coding sequences, those filled colored bars, are on one of the two strands, which is called a heavy strand, and a few of them are on the other coding strand. That one's called a light strand. So those proteins are mostly involved in electron transport and ATP synthesis. In addition to those protein coding genes, there are a couple of genes for ribosomal RNAs and some genes for transfer RNAs. However, there aren't any genes for ribosomal proteins. Those are encoded in the nucleus. So there are lots of proteins that are used in the mitochondria and that aren't actually encoded in the mitochondrial genome. They're encoded in the nucleus, and they have to be synthesized outside of the mitochondria and then transported in. So these are some of the proteins that are encoded in the mitochondrial genome, and these here are proteins involved in electron transport, and during electron transport, hydrogen ions are pumped outside of the cell, and then they come back through the ATP synthesizing enzyme, and that's how the ATP is made. So you'll notice that oxygen is used here as a terminal electron acceptor, and most of the oxygen that you breathe is for supporting your mitochondria. So if you stop breathing, your mitochondria will stop working and you'll stop working. So why do these organelles have a genome? Mitochondria have genomes because they used to be aerobic bacteria. They used to be free-living organisms. And about 2 billion years ago, these bacteria joined up with primitive nucleated cells to make the aerobic eukaryotic cells. The mitochondrial ancestors are related to bacteria called rickettsia, some of which now cause typhus. When the mitochondria became endosymbionts, that is, internal symbionts inside the cell, sorry, I lost track of what I was saying. When they became endosymbionts, they transferred a lot of their nuclei, or a lot of their genes, to the nuclear genome. So the mitochondrial genome is quite small, although in some organisms it's a lot bigger. For example, in the flowering plants, the muskmelon genome has about 2,400,000 kilobases of DNA, but mitochondrial genomes in plants are strange for ways that we're not going to talk about today. Another organelle that has genomes are the chloroplasts that are found in photosynthetic organisms. And both the chloroplasts and the mitochondria are produced by replicating themselves inside their host genomes or inside their host cells. And so they have a genetic lineage that's independent of the nuclear lineage. So nuclear inheritance looks like this. Organisms that reproduce sexually have two parents, a mom and a dad, and each parent supplies half of the chromosomes for each of their offspring. And for each kid, a new half from each parent is selected. So even though you'll share a lot of your genes with your siblings, it's only about half, and that's an average. So to get an idea of how many different ancestors contribute to your nuclear inheritance, if you go back just 6 generations, you have 64 grandparents. And that's more grandparents than you have chromosomes. So the contributions of any one nuclear, or the nuclear contributions of any one individual, get diluted out pretty rapidly. So all of your nuclear heritage comes from some ancestor, but not much of it comes from any individual ancestor. For example, this is a comparison of my own chromosomes with those of my daughter and those of a first cousin and those of a second cousin. So my daughter, of course, has half of my DNA, so she's got quite a lot in common with me. But if you start looking at the cousins, then that contribution gets diluted out even further because of crossing over during gamete formation between the parental chromosomes. And so these little bits are the part that I have in common with my first cousin, and these little bits are parts that I have in common with my second cousin. So the first cousin is sort of magenta, the second cousin is sort of orange. So you see that it dilutes out very fast when you're looking at the nuclear genome. In some cases, like this chromosome here, we have nothing in common, even though we're separated only by a few generations. The mitochondrial genome, on the other hand, is inherited only in the maternal line. So you have only a single lineage represented by your mitochondrial sequence. It goes down from great-grandmother to grandmother to your parent to yourself, to your mom, to yourself. So it's only transmitted in the female line. So all of your mitochondrial DNA comes from a single ancestor. So my daughter and I and my first cousin both have the same mitochondrial DNA because we have a common maternal grandmother. However, my first cousin is male, and males don't transmit mitochondrial DNA. My second cousin's mitochondrial DNA is different from her father. So only a single individual contributes mitochondrial DNA to any individual, and the difference between mitochondrial DNAs has risen only by mutation over the generations. So each group of descendants of a given female are going to have similar mitochondria. And that brings us to mitochondrial Eve. In 1987, in January, Rebecca Cann and Mark Stone King and Alan Wilson published an article in Nature, and it was called Mitochondrial DNA and Human Evolution. And this article described their comparison of the mitochondrial DNAs collected from the placentas of 147 women from different parts of the world. That is, the placentas that they were carrying for their offspring. Donated their placentas, and the mitochondrial DNA was extracted from those. And they were from Africa, Asia, Europe, Middle East, Australia, and New Guinea. So in this sequence, they did not sequence the entire mitochondrial genome. They didn't look at the whole 16,000 plus bases of the genome. Instead, they were looking at what's called restriction sites. And these are little short bits of DNA that are recognized by enzymes that cut the DNA at these specific short sequences. Most of these sequences are about 6 bases long, more or less. And they read the same in both directions, so they're palindromic, like ACCGGT. And because of the base pairing rules, with A matching with T and C matching with G, if one of the two DNA strands reads ACCGGT, then the other strand, reading in the opposite direction, also reads ACCGGT. The restriction site has that peculiarity. So they found 133 different sets of restriction sites in their subjects, each of which was found only in one geographic region, although some of the sets were shared by individuals in the same sample set. And ultimately, these kinds of differences were called haplogroups. So Can, Stoneking, and Wilson made this big tree of mitochondrial sequences that showed how the different mitochondrial sequences in their samples were related to one another. And since differences in the mitochondria arise only by mutation within whatever lineage they're in, then each lineage accumulates different mutations. So groups of individuals that have a similar set of mutations were eventually called haplogroups. So they identified two major branches of the tree. I don't know if you can see these little symbols here, but these represent the countries that the different mitochondria came from. And the African symbols are filled circles. So they found that one of the two branches consisted only of African sequences, and the other main branch consisted of everybody else, of some African and some non-African sequences. And since the sequences within the African branch showed the most variation, and they had accumulated more mutations than any other group, Africa was considered to be the origin of the oldest group of mitochondria. And the mitochondrial ancestor, the common ancestor of all of the mitochondria, was placed at the midpoint between those two major branches. So based on the assumption that there was a mutation rate somewhere between 2 and 4% per million years, they calculated that the mother of all mitochondria, arose in Africa between 140,000 and 209 years ago. So that concept became known as mitochondrial Eve, that concept of an ancient maternal mitochondrial ancestor. And it was natural to associate that mitochondrial ancestor with the biblical Eve. And in October 1987, Roger Lewin, who's a science writer, reported the work in Science using the reference term mitochondrial Eve. He wasn't the first person to associate the common mitochondrial ancestor with the biblical Eve, but I think he was the first person to use that. So he was quick to caution against taking that association too literally. It's not that all living humans are the descendants of a single human couple or even a single human woman. So what mitochondrial Eve represents is a matriline, a matrilineal lineage of mitochondrial descent. Oops, sorry. There we go. Okay, so what this sequence represents, or what this ancestral sequence represents, is the persistent mitochondrial lineages, because some lineages disappear and others don't. We'll see why in just a minute. So that persistent mitochondrial lineage was founded in Africa about 160,000 years ago. One question is did that sequence belong to an early modern human? Maybe, maybe not. Early modern humans arose in Africa around 200,000 years ago and started migrating into Asia and Europe about 100,000 years ago. And a lot of the genes in a nuclear genome existed before modern humans, and so the mitochondrial genome also existed before we emerged as a species. So whether it was one of our early modern human ancestors or somebody before that, we're not quite sure. So what this shows is the inheritance of mitochondrial lineages. So you only get them from your mom. So this one went on from mother to daughter to daughter to daughter to daughter. And so there are a lot of those still present, but other lineages can die out. For example, if you pass a mitochondrion to your son, that one doesn't go on to its progeny. Male children don't pass on their mitochondria. So it's possible for a lot of mitochondrial lineages to become extinct. We're going to see an example of that a little bit later. But for right now, for example, I have one daughter, but she has no children, and my sister had 2 boys and a girl, and the girl in turn has a boy and a girl. So our particular mitochondrial lineage is riding on that particular grandchild's reproductive success. But the idea of mitochondrial Eve really got into the public imagination. In fact, it may even been of the first instance of using the term DNA to represent the coding of various cultural practices. You hear a lot that a particular idea is in the DNA of a particular group. And so I think maybe this idea of mitochondrial Eve might even have been the first instance of using DNA just to represent various attributes of different cultures. But in 2001, Brian Sykes wrote a book called The 7 Daughters of Eve. And in this book, he describes 7 European and Middle Eastern branches of the mitochondrial tree, and he assigned each of them to a female ancestor who was given a name that matched one of the haplogroup designations. And these are those 7 haplogroups: H, X, U, K, T, J, and V. And so the women that were named for those were Helena, Xenia, Ursula, Katrine, Tara, Jasmine, and Velda. And so he told a story about, a plausible story, about each of these individuals. And you'll notice that most of these are fairly young haplogroups. Like the oldest one is Ursula here. That only goes back about 45,000 years. Another thing that was in the book was the story of the discovery of Otzi the Iceman. This is a passage from Sykes' book that describes the discovery of a 4,000-year-old human body frozen in alpine ice. On Thursday, the 19th of September 1991, Erika and Helmut Simon, 2 experienced climbers from Nuremberg in Germany, were nearing the end of their walking holiday in the Italian Alps. The previous night, they'd made an unscheduled stop in a mountain hut, planning to walk down to their car the next morning. But it was such a brilliantly sunny day that they decided instead to spend the morning climbing a 3,516 meter Finalspitze. I hope I said that right. I probably didn't. But on their way back down the hut to pick up their rucksacks, they strayed from the marked path into a gully partly filled with melting ice. Sticking out of the ice was the naked body of a man. So we'll hear more about Otzi later. Now, the idea of 7 daughters of Eve was a compelling one, but there are way more than 7 haplogroups. The ones that were mentioned in the Sykes books are circled here. These are major European and Middle Eastern haplogroups, but you can see there's a lot of other different haplogroups. This whole bunch here, for example, all of these guys here. All of the L haplogroups are African lineages, and haplogroup L3 represents the group that migrated out of Africa, and that was subdivided into two subgroups, M and N. Each of those was subdivided into several haplogroups, like N divided into R, AS, V, and X. My particular haplogroup is I, and this came from IWX, which came from N, which came from L3. One of the things that mitochondrial DNA is useful for is tracking migration pathways. And this shows the migration of L3 out of Africa. Here's the N and M subgroups, N giving rise to R giving rise to a bunch of others, N giving rise to a bunch of others, M giving rise to a bunch of others. And they all migrated out to other parts of the world. And some of the eastern haplogroups, the ABCD haplogroups, migrated across what was the Bering Land Bridge into New World in the Americas. And so a lot of Native Americans and their Hispanic descendants are haplogroups A, B, C, or D. Now, the first mitochondrial comparisons, the one by the Can, Stoneking, and Wilson report, did not sequence the entire mitochondrial genome. The first, let me skip that. The first full mitochondrial genome that was sequenced was done by the Sanger lab in Cambridge, and they did the full mitochondrial DNA sequence, all 16,000 plus bases, of an unidentified woman of European descent, and this went into the GenBank database and came to be known as the Cambridge reference sequence. There were some errors and some inconsistencies in that sequence, and those were later corrected and published by Richard Anderson as the revised Cambridge reference sequence, or the RSRS. But the RSRS is now the sequence that's used as a reference sequence for defining mitochondrial variants. And this is part of the Cambridge reference sequence. This is the part of the sequence called the control region. Called the control region because this is the point of origin for replication of the mitochondrial chromosome. It contains two large hypervariable regions, HV2 up here, HV1 down here, HV3 here. The HV, of course, is for hypervariable. And each of these contains a number of mutations that can be associated with various mitochondrial haplogroups. These are frequently studied because they're hypervariable, because they accumulate a lot of mutations. And they can accumulate a lot of mutations because this region doesn't encode any of the mitochondrial proteins. So mutations in that region don't result in defective proteins and non-functioning mitochondria and do damage to their owners. So there are mitochondrial defects that can damage their owners. For example, Leiter's hereditary optic neuropathy lead to blindness. And recently, Chantal called my attention to an article about a woman whose longevity was attributed to the endurance of her mitochondria. We've been hearing recently about Donald Trump's excellent health, and I'm wondering if Trump's doctor's assessment of Trump's health is accurate. Maybe he has good mitochondria too. But unfortunately, since he's a male, his children can't inherit them. So the mitochondrial haplogroups are defined with respect to the revised Cambridge reference sequence by specific mutations, some of which are in the coding region, and some of which are in the hypervariable region I showed you. So these are the defining mutations of some of the haplogroups, and it lists only the mutations of the major group. Each of these has a number of subgroups. And sometimes those subgroups go into multiple letters and numbers. But these are some of the major haplogroups. It's just a few of them. So this is just an example of how the haplogroups are defined. So what you have here is a letter and a number and another letter. And the first letter is the base that's in the Cambridge reference sequence. The end letter is the base that's found in the haplogroup. That's the change in the haplogroup. And the number is which of the bases has been changed. So this gives you a reference position for each. And sometimes they change back. This exclamation point here represents substitution that go back to an ancestral base, not from the Cambridge reference sequence, or not necessarily from the Cambridge reference sequence, but from whatever the preceding branch of the mitochondrial sequence was. This is a family tree from student mitochondria. Every year in a lot of our biology classes, we do the mitochondrial DNA. We do one of the hypervariable regions, usually HV1, for the mitochondrial DNA of our students, and we have them sequenced. And we usually do a tree for the class so they can see how their mitochondria are related to one another. You can see there aren't many differences. We have at most 7 differences between the mitochondrial DNAs of the students in the hypervariable region. And since HV1 is only about 350 nucleotides long, then that's why there's just a few percentage points differences between those different samples. But it's fun to do, and the kids get some idea about their mitochondrial lineage, where the maternal ancestors came from. And it's fun for them, and it's fun for us. Okay, so back to mitochondrial Eve. You may have already correctly inferred that the sequence of the Cambridge reference sequence is not the sequence of mitochondrial Eve. Mitochondrial Eve is also known as the matrilineal most recent common ancestor, or the MMRCA. And the revised Cambridge reference sequence is a European sequence that has the haplogroup H2A2A1, so it's one of the many subgroups. And all of the European sequence, all of the H group sequences are more recent, much more recent than the MMRCA, the common maternal ancestor. So in 2012, there was a coalition of scientists, Doran Bayar and Manus van Oven, and a bunch of others from several different countries, that proposed what they called a Copernican reassessment of the reference mitochondrial sequence. And what they wanted to do was to shift the reference sequence from its Eurocentric position to what they called a phylogenetically valid reference point. And they collected almost 19,000 complete mitochondrial DNA sequences and 6 Neanderthal sequences to use as a reference point. And from those 18,843 sequences, they were able to infer the ancestral sequence, or the sequence of Eve. And that sequence fell between haplogroups L0 and L1. That sequence is right here. And so it falls between L0 and L1, which are both African sequences. And they differ from each other by about 14 mutations in the coding region and 4 control substance regions relative to the reference ancestral sequence. So the Cambridge reference sequence had become the reference sequence because it was the first mitochondrial genome sequence to be published. But by 2012, there were a lot of mitochondrial sequences, that is, over 18,000 of them, available in the databases in addition to those 6 Neanderthals. Now, it's interesting, or maybe not too surprising, that the reconstructed sapiens reference sequence, or the RSRS sequence, falls very close to where the originally reported ancestral root was between haplogroups L0 and L1. African, but it differs from both of them. So this is the control region of the RSRS, the reconstructed sapiens reference sequence, and each of these capital letters in red represents a difference from the Cambridge reference sequence, although the reference numbers here are the same as the numbering of the Cambridge reference sequence. So they kept those reference numbers because the haplogroups are defined with respect to those numbers. So they thought it would be just too confusing to change the numbers to match the RSRS. So they didn't do that. There's about 20 differences between the Cambridge reference sequence and the RSRS, and there's been some discussion in the literature about whether switching to a new reference sequence for defining the haplogroups would be confusing, and some sequencing services cover all their bases by just reporting both, saying that you are different from the Cambridge reference sequence at these points and you're different from the RSRS at these points. However, changing the reference sequence doesn't change your haplogroup. It just changes how that haplogroup is defined. So the Copernican group also worked out a big mitochondrial tree for all of their sequences, and you can see it gets pretty solid near the right-hand edge here. But they calculated that the revised sequence arose about 177,000 years ago, and that L0 and L1, respectively, rose about 136,000 and 128,000 years ago. So it was much later. L3, which is a group that left Africa, rose about 67,000 years ago. And then the two groups that diverged from L3 arose about 49,000 and about 59,000 years ago. So those are some of those major branch points. And then the revised Cambridge reference sequence is way over here. So it's very revised. So it's definitely a European sequence. But we'll see whether the RSRS becomes the new reference sequence or whether the Cambridge reference sequence continues to be used just because it's been used for so long. So now we're going to go back to Otzi. So Otzi's body was freeze-dried so that both his nuclear and mitochondrial DNA could be isolated from it. His age was determined from the tools that were found with him to about 4,000 years ago. He was apparently killed by an arrow. His nuclear DNA was checked and he had blood type O, and he was lactose intolerant, and he might have had heart trouble if he hadn't been killed by the arrow. And his mitochondrial haplogroup was type K, which is a common European haplogroup. But his subgroup was K1F, and there are no living relatives of K1F in the databases. So they thought, well, maybe somewhere in the region that Otzi was discovered, we might find some living relatives. And so they collected mitochondria from a bunch of people in that region around the Italian Alps. So those mitochondria were all sequenced, and Otzi's genetic structure was compared with all of those. So these are the neighborhood haplogroups. And I don't know how easy this is for you to read or not, but this is the haplogroup U. This is the haplogroup K that's a descendant of U. This is the haplogroup K1. And then here are several of the subgroups. So we have K1D, we have K1F, and we have K1E. Here's K1D1. Here's K1F. Sorry, K1E, K1E1, but there aren't any other K1Fs. So Otzi is the only remaining exemplar of his particular haplogroup A1F. And that just shows, I mean, he's only 4,000 years old. So that just shows how easily a particular mitochondrial lineage can get lost. So we've gone from so-called mitochondrial Eve about 177,000 years ago to Otzi about 4,000 years ago, and all of you also have mitochondria. And do any of you know what your haplogroups are? So Mike, you had yours done by National Geographic Sequencing Service? I had mine done at 23andMe. And there's a lot of sequences, a lot of people that know what their haplogroup is. A friend of mine teaches anthropology, and they ran their mitochondrial DNAs and got their haplogroups. And after they got their haplogroups, they had all the people with the same haplogroup sit together as a group. So they used their DNA to group them within the classroom. Well, it's kind of fun to know. So if you ever get a chance to get your mitochondrial haplogroup checked, then do. So you'll know where you are on the mitochondrial tree. It's kind of fun to get a haplogroup designation. Let me just run back. I skipped over this a minute ago, but let me just run back to this one. Back a little further than I thought. There it is. Oh, this is my haplogroup. My haplogroup is I2, and the I haplogroup is the subgroup of N. And the I haplogroup appeared about 21,000 years ago. It's actually a very minor haplogroup. There aren't that many people. And if my family's reproductive patterns are any indication, I can see why there are so few of us. Psychically connected. Interesting. No, mother and daughter cannot have different haplogroups. You're going to get your haplogroup from your mom. So if you know what your mom's haplogroup is, you know what yours is. So my mom had the same haplogroup as my grandmother, and my first cousin, who is the son of one of my daughter's or one of my mother's sisters, has the same haplogroup that I do. So we're all in the same lineage, but there aren't that many female offspring in the family. Yes, that timeline does bring home how new humans are. So eventually my female ancestor wound up in Ireland, which makes sense because that's where my maternal grandparents came from. Yes, so any change that gets made in the in the sequence has to be made by mutation because you don't get any mixing from parents. Every now and then you'll get a male mitochondrion into an egg, but generally because it might be just one and there are lots of mitochondria in the egg, it tends to not to be transmitted. But it's unusual to get male mitochondria transferred at all. They tend to only the nucleus gets into the cell. So you don't get mitochondrial mixing because it's a straight line inheritance. So it's very nice. It's kind of like the Y chromosome in males. You have a Y chromosome, you got it from your dad, who got it from his dad, who got it from his dad, and so forth. So there are also Y chromosome haplogroups that are defined, so you can track both your paternal and your maternal. Well, that's an interesting question because there probably are a lot of haplogroups that have been lost or have been lost simply because female only had male children, and so her lineage was not passed on. So the haplogroups that we have are the surviving haplogroups. And there's a bunch of them. I'm sorry, okay, I'm back. Um, Let me see, I think I lost. Okay, so far as I know, there isn't any correlation between mitochondrial haplogroups and any any health issues, for example. There are health issues that are associated with mitochondria, but I don't think they're specifically haplogroup associated. I'm not really good at keeping up with the chat, so is there any questions that I didn't address that are a little further back? I don't see anything, but if there's a question you had that didn't get answered, please feel free. Well, thank you all for coming. Um, I don't do these very often, but I always enjoy it when I do. And get your mitochondrial haplogroups. It's fun to know these things. Oh, okay, let me see. Oh, does mitochondrial DNA have native viruses? I have no idea. That is a really, really good question because bacteria certainly have viruses. Um, and the bacteria that the mitochondria were descended from probably had viruses. It's a really good question. I don't know. It is a little bit more prone to mutation because the proofreading of the mitochondrial DNA polymerase is not as good as it is in the nuclear genome. Haplogroup divisions. Oh, okay, sure. I think you want this one. Is that the one you want? Is there anything else you want to see again? Oh, a cloning of mitochondrial DNA to preserve a lineage. Oh, you probably know that there have been some mitochondrial transfers from people who have healthy mitochondria to people who don't, and they don't want to pass them on to their children. They might have, some kind of mitochondrial disorder, and so normal mitochondria have been inserted into an egg that is then in vitro fertilized. And yeah, so you've got actually three parent zygotes. Males have mitochondrial DNA. They get it from their moms, but they don't transmit it. Apparently, and there was a huge amount of ethical discussion about whether or not that was a good thing to do. Yes, as a matter of fact, if you go to Genome Island, there in the tower, there is a link to that story about three parent babies. Mitochondria are the most suitable target of cells under attack from microorganisms like viruses or ROS produced upon different viral infections. Huh, I'll have to check that out. Oh yeah, well that's true. Well, thank you, Chantal and Jess, for inviting me to do this. It was fun putting it together. Actually learned a few things I didn't already know about mitochondria, so that was great. Well, I loved being here. Um, yeah, telomeres are interesting. Telomeres are put on separately from the rest of the chromosome. They're not encoded in the genome. They're actually attached by an enzyme, a telomerase. And the ends of linear chromosomes tend to get reduced at every replication cycle, and ultimately you start to get down to the ends of chromosomes that have genes that you don't want to throw away. And unfortunately, those telomerases stop working as you get older. So mine have probably been dead for decades. And so the telomeres do get reduced a little bit every time your cells divide. Well, thank you, Heron. Oh dear. Film. All right, that's great. Thank you. Oh, yes. Yes, I read about that. Okay, I'm going to turn the voice off now and slurp some more coffee. Thank you all.